Hello, my name is Evelyn McChesney.
And my name is Madeline McCue, and the two of us turned bacteria into antibiotics. So over the past 40 years, there's been an influx in something called antibiotic-resistant infections. These are normal bacterial infections that have mutated to gain resistance to the antibiotics we would use to treat them. These antibiotic-resistant infections are going to be a huge problem. By 2050, they are predicted to kill 300 million people, about the population of the US currently, and cost the global economy over $100 trillion.
So the purpose of our study was to engineer a novel alternative to antibiotics by engineering probiotic bacteria to produce a type of protein called antimicrobial peptides. Antimicrobial peptides are small proteins naturally secreted by bacteria and the human immune system. In bacteria, they're used to target other bacteria. And in the human immune system, they're used to fight infections. However, they aren't usually produced in high enough quantities to treat most bacterial infections. Therefore, the goal of our project was to harness the ability of bacterial AMPs to fight infections to augment the human immune system.
Now, we were really interested in AMPs because of the way they work. Most AMPs target the cell membrane of the bacteria, and bacteria have a really hard time rapidly changing their cell membrane, so it's hard for bacteria to develop resistance to them. So AMPs work because they're both lipophilic, attracted to the lipid membrane, and positively charged. So an AMP will gather at the lipid membrane. As more gather, since they're positively charged, they will repel each other, and that will start to tear the membrane open. As more gather, they will open the membrane even further, creating a pore. We call that pore a micelle. From there, water that's outside the cell will flow into the cell. This increases the intracellular pressure until the cell physically explodes.
There are some problems with using AMPs currently. The first is that they can't be injected into the bloodstream because they'll be attacked by white blood cells. Second, they can't be ingested orally because they'd be broken down by the enzymes in the stomach.
The other issue with AMPs is that they're pretty specific. Most AMPs target only gram-positive or gram-negative bacteria. There are two types of bacteria, these gram-pos, gram-neg, and they're differentiated by their cell membranes. And since AMPs target cell membranes, you have to have different ones to target different kinds of cell membranes. Last year, we solved this problem by engineering two separate systems, one that targeted gram-negative bacteria and the other that targeted gram-positive bacteria. However, when a patient comes into a hospital with an infection, they don't typically know what kind of bacteria they're infected with. So we wanted to create a broad spectrum treatment, something that could be given to any patient with a bacterial infection that would work.
So this year, we chose to engineer probiotic E. coli Neisel 1917 to produce two AMPs, one to target gram-negative bacteria and one to target gram-positive bacteria. To first create this design and engineer this bacteria, we created a digital design of a piece of DNA called a plasmid. And this is the design that we use to engineer all of our research.
So once we finished our digital design, we started our wet lab work. So the first part of that was to create a base plasmid, PMKP+. PMKP+ would have two primary components, the first of which would be the plasmid backbone, PMKRQBB. We selected this plasmid backbone because it has 5 restriction sites, places where we can easily cut and paste DNA. The second primary component was the Proteon Plus promoter. This is a strong, self-regulating promoter that will encourage the bacteria to produce our DNA in high quantities.
To begin engineering this base plasmid, we first had to isolate that Proteon Plus promoter. Based on our digital design, this was predicted to be 1.3 kilobases, which we confirmed using gel electrophoresis.
So from here, we had to insert that Proteon Plus into PMKRQBB to get our base plasmid. We inserted it using 2 restriction sites and enzymes, EXPO1 and KPN1, to create, there we go, PMKP+. We used gel electrophoresis to confirm that this worked. Using colony PCR, we predicted on our digital design that this would be 1.6 kilobases, and our green line up here shows that it did indeed work. So we've got our base plasmid.
The next step in our digital design was to choose which AMPs we wanted to use to target bacteria. We chose microcin L based on a study by Pons et al., which suggested that microcin L is active against gram-negative bacteria, and we chose entericin A based on a study by Americk et al., which suggested that entericin A is active against gram-positive bacteria.
Now, if we want to get our bacteria to produce these 2 AMPs, we have to engineer a whole nother portion. We're calling this secretion machinery, a pathway of proteins that will allow the bacteria to transport our AMP out of the cell membrane and where it can start doing some work. So we took a look at the microcin L secretion machinery. However, this would have been perfect because it would match the microcin L AMP, but this was very large and very expensive and would have taken a lot of time to get. So instead, we took a look at the microcin V secretion machinery. The microcin V secretion machinery was available to us in our lab, and it was highly homologous to the microcin L secretion machinery.
One problem with using the microcin V secretion machinery is that this secretion machinery is found in the PHK22 plasmid. And also in this plasmid are the CVAC and CVI genes. CVAC encodes for a protein that we did not want to be produced when we isolated this secretion machinery. So we decided to use site-directed mutagenesis to change the start codon in front of CVAC to a stop codon to prevent the bacteria from reading the CVAC gene and thus preventing the production of the unwanted protein.
To start this process, we took that chunk of DNA that CVAC and CVI are on, and we split it into two pieces, fragment A and fragment B. We mutated both of these pieces using a process called site-directed mutagenesis, and confirmed that this isolation worked using gel electrophoresis.
The next step was to combine fragments A and B into fragment C. This fragment is exactly the same as the fragment we took out of PHK22, the plasmid with the secretion machinery, except for it had the stop codon instead of the start codon, so it would prevent the encoding of the CVAC gene. And this we confirmed with gel electrophoresis based on our digital design.
Following this, we had to reinsert that fragment C into PHK22 to get PHK22 delta. This would be identical to the original PHK22, except it would not produce that protein encoded by the gene CVAC. Here we go. We used gel electrophoresis to confirm that it worked, and it did.
So the next step was to isolate this mutated secretion machinery, and which we predicted was 9.2 kilobases based on our digital design. And this was successful, and we proved this with gel electrophoresis.
From here, we have to insert that isolated secretion machinery into our base plasmid. Using restriction sites PAC1 and SPE1. This was our ideal plasmid, PMK P plus V. However, that secretion machinery is 9.2 kilobases. For those of you who are familiar with genetic engineering, that is a really, really large chunk of DNA. So this was a very difficult process. We spent many weeks on it, but we eventually ran out of summer. However, one of the grad students in our lab was really interested in our work, so she picked up our work, and her results will be published soon.
So although we didn't finish our laboratory work, we did finish our digital design. So the next step was to engineer the gene blocks, the fragments of DNA that would encode for the AMPs. So each gene block has an AMP and its self-immunity gene, so when the AMP was secreted, it wouldn't kill the bacteria that produced it. A secretion tag so that the secretion machinery would recognize the AMP and bring it outside of the cell. A multiple cloning site and a terminator sequence. The multiple cloning site allows us to add more genes in the future so we can produce more AMPs from one plasmid. And a terminator sequence stops the reading of that gene. There's also two restriction sites, SAC1 and PAC1, which allow us to insert this piece of DNA into our base plasmid that also has the secretion machinery.
For the secretion tags, we selected three. The microcin L secretion tag, which would match the microcin L AMP, the microcin V secretion tag, which would match the microcin V secretion machinery, and YEB-F, a general secretion tag for E. coli that would match our delivery vehicle. All in all, we created three sets of gene blocks. Each set would have a gene block that encoded for microcin L, and the other would have a gene block that encoded for enterocin A, excuse me. Each one would have the microcin L secretion tag, the microcin V secretion tag, and the YEB-F secretion tag.
In the future, we want to insert these gene blocks into the base plasmids with the secretion machinery, then insert these final plasmids into our probiotic E. coli, and analyze which probiotic E. coli produces enterocin A the best. From there, we can isolate that enterocin A gene block and insert it into the multiple cloning site of the microcin L plasmid that produces microcin L the best. This would give us a broad spectrum plasmid that, when inserted into E. coli Neisel, would be able to target both gram-positive and gram-negative bacteria. We would then analyze these results.
So, in conclusion, we created a complete digital design for a genetic system that would turn a common probiotic, it's FDA approved, E. coli, into an antibiotic by producing two antimicrobial peptides. In the lab, we began the process of replicating this digital work. We isolated the proteon promoter and inserted it into PMK RQBB to create a base plasmid. From there, we took the microcin V secretion machinery, isolated it, mutated it, and inserted it back into PHK22. From there, we isolated that large chunk and began the process of inserting it into our base plasmid. Although this work wasn't entirely finished, it's an important step in the process of generating a new type of antibiotic that can save some of those 300 million lives that would otherwise be lost to antibiotic resistance.
We would like to acknowledge our advisor, Dr. Yiannis Kaznessis, 2 of the graduate students in our lab, our teacher, the Siemens Competition and Discovery Education.
Here is our obscenely long list of references. Thank you very much.